Welcome to our first lecture in Bio 4B. We're going to take a look at the history of life on Earth. And so one of the big things about this particular topic and chapter is, if you think back to like astronomers and so forth, right? The universe basically started with the Big Bang Theory. That's what's hypothesized and so forth. And it's been pretty well supported. But, life on Earth, we can't travel back to take a look at what was there. So we're relying upon things like fossil evidence and, taking core samples of ice and soil and down deep in the oceans and so forth. We know locally, if you ever hiked up in the Santa Ana Mountains locally, you'll find shells up there. Right? I have some slides that I'm going to talk about when we go out to the deserts. Oh, well, I'm not going to take you guys out to the deserts, unfortunately. But if you go out to the deserts, our local desert out in the Mojave Desert, you actually find trilobites. So all that stuff was under water and so forth. So we have evidence to support all this. So in this particular chapter, what we're going to do is we're going to take a quick little trip and talk about the history of life on Earth for animals and so forth. Okay, and then so on the slide right here, what you're looking at is essentially your study guide. These are the generalized topics that I'm going to cover with you in this particular chapter. So in this lecture and then also in the next lecture as well. As you go through this topic, if I spend a lot of time talking and explaining things over and over and over again, that's probably a good short answer question. But what I'll do is I'll actually tell you guys here are the potential short answer questions. And typically when you're in my class, I will always, on the test, right? If we have 4 chapters on the test, I will always ask you a short answer question. One question out of each particular chapter on there. And I want you to be able to explain to me what you know. Okay, so that's the way I tend to operate. But here's the generalized study guide. Now you see the people down here at the very bottom down here. I do expect you guys to know some folks, and we'll talk about some of these folks as well. So let's get started. So the first thing we're going to take a look at is talk about the early atmosphere. And in the early atmosphere, here's this nice big list of molecules that were present, and one of the things here is when conditions are ideal, potentially these molecules are going to be able to be synthesized together. And when we take a look at these, we're going to talk about the abiotic synthesis of organic molecules. And the early organic molecules that were synthesized were most likely nucleic acids and amino acids, right? Or the basic building block of nucleic acid, nucleotides are the earliest ones. And once we have the basic precursors, right, these monomers that you guys are all familiar with, the nucleotides and amino acids can be synthesized together to form macromolecules. And so nucleotides are going to be used to make nucleic acids such as DNA and RNA. Amino acids are going to be synthesized to make polypeptides. And so you guys are probably already all familiar, or hopefully you should be all familiar with all the various functions of nucleic acids and proteins. Now that macromolecules have been formed, the next step in this is the formation of protobionts. Protobionts are essentially a lipid enclosed, it's a lipid membrane that's kind of enclosing these various polymers. So the nucleic acids, the polypeptides are enclosed within a little sac. So almost like a vesicle or a vacuole. Right? And so if you've ever taken oil and you drop oil into water, right, and you mix it all up and you see all of them, they kind of break apart, and then all of a sudden they start to reform these larger vesicles. So that's what we're taking a look at with these protobionts. But now that we have protobionts that contain these polymers inside, the next step is what's the mechanism for self-replication? How did the self-replication come into play? And so when we take a look at the various hypotheses that are out there, where some of these organic molecules came from and where these organic molecules were to allow for the synthesis of these nucleotides and amino acids, which then allow for the formation of macromolecules, which then allow for the formation of protobionts. And then these protobionts now were then able to somehow self-replicate. So the idea where a lot of this stuff came from is, there is the extraterrestrial hypothesis, and we do know this, and I'll talk about this in just a little bit. We do know that meteorites, there have been polymers found on these meteorites. If we look at volcanic activity, we know that in the early Earth history there's a lot of volcanic activity. So if you're ever driving up to Vegas, there's actually a lot of cinder cones out there. So if you look out off away to the east and west of I-15, especially if you get out towards Baker, right? That's where the world's tallest thermometer is. If you look east, you'll see these little brown cones out there, and those are actually cinder cones. If you've ever driven up to Mammoth, once you get past when you get up there near the sign that says Fossil Falls, you see this big old red cinder cone sitting right there. That right there, that's an ancient volcano. And then we have these hydrothermal vents also that allow for some of these molecules to be released. So back in the 20s, two individuals, Oparin and Haldane, these guys independently proposed an idea, or they hypothesized that in order to form these biologically important molecules, the atmosphere must have been a reducing atmosphere. So this became known as the reducing atmosphere hypothesis, right? And so in order to go from monomers to polymers, you guys all know that you have to supply some energy, right? So in order for you to grow, you have to have some energy available to allow for the proteins to put the monomers, to link them together via dehydration synthesis. So early on, what was the idea, right? Where the energy came from? Well, there was a lot of lightning, the sunlight, UV radiation, and volcanoes. And one of the big things about the volcanoes is we do know that with volcanoes, right, they spewed out a lot of carbon dioxide, a lot of nitrogen. Notice that our atmosphere is made up primarily of nitrogen gas, right? 78% of it's nitrogen gas. Right? And so now all of these molecules that you saw earlier on the list, the ammonia, the methane, the carbon dioxide, the nitrogen, hydrogen sulfide, and so forth, all those things form that primordial or that primitive soup. And based upon where these molecules were, if the conditions were ideal, then they might be able to form together. So in the 50s, Miller and Urey, Stanley Miller was actually the graduate student in Harold Urey's lab. What they proposed, or what they actually did, was they set up an apparatus where they tested the reducing atmosphere hypothesis. So here's their water sea, right? They had heat kind of generating water vapor coming up, and up in the atmosphere they had methane gas, they had ammonia, they had hydrogen gas up there as well. One thing that they really didn't put in there was hydrogen sulfide, and that's going to be important a little bit later on. And so to supply the energy to allow these molecules to kind of link up together to form bonds, they used an electrode to generate some electrical activity. And then when the atmosphere cooled and everything condensed down, and they sampled their chemicals, what they found was formaldehyde. They found hydrogen cyanide in their sample. So they found some organic molecules that weren't present over here, right? And so they are actually able to find some amino acids. So if we jump forward to to the early 2000s, I guess 2010, 2011, right? Jeffrey Botta down at Scripps University, like, let's test Miller and Urey's hypothesis. And so what he did and his group did was they actually were able to find additional amino acids, 10 more than what Miller and Urey were able to discover. And they also found they were able to synthesize 4 amines from it. So that was completely different from what Miller and Urey found on there. Okay, and so what Bada actually did was it actually kind of came back and more with different types of equipment, kind of was able to test that reducing atmosphere hypothesis. Now, part of the reason why Bada ended up with additional amino acids and so forth is he tested hydrogen sulfide. One of the reasons why they speculated that Mill and Urey probably didn't work with hydrogen sulfide is, I mean, you guys have all in chemistry, right? Probably there is that lab that deals with hydrogen sulfide. I remember as an undergrad, right? It's like, all right, about week 8 in the quarter, it's like, oh man, the hallways stink. The heck? Who had eggs? Who had? Oh man, somebody checked their drawers. So what happened is, you know, that's probably the reason. So what Bada actually did, these guys did, they actually tested out. They actually used hydrogen sulfide, and that's how they were actually able to come up with the addition. So if you want to read more about this, you can kind of click right there, and you can find out more about that. Right? And so now there's this other idea that the atmosphere, maybe it wasn't a reducing atmosphere because there's carbon monoxide, CO2, nitrogen gas, right, and water. Those are all pretty neutral, and we get a lot of this from volcanic activity, from volcanoes spewing all that stuff out. And so now there's kind of, I don't know. So the other two hypotheses that folks actually went out and tested to look for evidence is the deep sea vents versus the extraterrestrial hypothesis. So in that very first image, right, you just saw the volcano, you saw, you know, the deep sea vent, and then you saw the comet coming in, right? So that's the extraterrestrial one. So with the extraterrestrial one, what are we looking at? We're talking about meteorites and asteroids and comets and so forth. And so whenever they hit Earth, they're going to bring in some additional polymers on there. And so what Bada actually did was they compared their hydrogen sulfide samples with those found on meteors, and they were very similar. Now, when we take a look at the deep sea vents, we're talking, you can see all along through here, right? Right along through here. So if you look right here, we're sitting right about here, right? Notice that we have all this red kind of it comes all the way around to this other side, right? That's the ring of fire, right? That's not what Johnny Cash was singing about. But at least I don't think so. So, but right there, this is what we're looking at. There's a lot of volcanic activity. Some of you guys are probably familiar with the Mid-Atlantic Ridge, right? So these are all these areas where they're sampling. And one of the things here is when the lava and this molten stuff is coming out, it's about 300 degrees Celsius. But the surrounding water is a lot cooler, so there's a gradient area where they're actually looking for a lot of these samples, right? So where we have the volcanic activity where it's releasing a lot of nitrogen gas, right? That can get synthesized into ammonia, and then from the ammonia that can be synthesized into amino acids and nucleic acids, right? So for those of you guys that have already had your chemistry, right, or even your first semester gym bio, right, there's your amine right there. In order for the polymers to be formed, we have to have ideal conditions. So it was proposed, and it's been speculated that in order for the polymers to form, it must have been some type of, you know, it wasn't dry, it couldn't be super wet. Although from some water samples now, they've actually found evidence of that. And that's where the hot thermal vents come into play. So not right at the hot thermal vents, not right where the cold water is, but there's a little gradient area where the water is a lot cooler. On the slide earlier, I didn't mention it, but about 50 degrees Celsius, right? So that seemed to be kind of idea to allow for polymer formation. And then in certain types of clays and muds and inorganic crystals. So here, if you look at this figure right here over time, when the monomers were present and it was just molecules only, they did not find very many of the polymers. But however, if they put it on a particular clay soil condition, right, the number of precursor molecules that were able to actually form the polymers actually increased in size. So now that we have polymers, now what we can start to form are these protobionts on there. So here you can see these protobionts. All right, so here's a large protobiont. If you look on the inside, it looks like they're actually reproducing. So this outer portion of this protobiont, that's the membrane right there. And then and then the internal, you can actually see some of the polymers inside, or at least, you know, you can pretend there. So now we need to talk about some catalytic activity. In order to link these things together, right, we have to have something that kind of helps to speed up the reactions. And so now when we're taking a look at this bottom part, I kind of pointed this out earlier, it kind of looks like reproduction. So when we look at Volvox, that's a protist, that's a green algal protist that we're going to take a look at. This right here looks exactly like Volvox. You have this large Volvox, it's a colony of a bunch of organisms, and they'll actually form little daughter colonies on the inside, and these are asexually reproduced. But with these protobionts, they have coacervates and they have liposomes. So when you take a look at these coacervates right here, so these coacervates, they actually, you know, they have these proteins on the inside, and protein nucleic acids on the inside. And so one of the things here is, here's this kind of like this primitive cell. Then liposomes, I kind of mentioned to you guys earlier, right? If you ever drop phospholipids into aqueous environment, they'll actually form little micelles. So some of you guys are actually probably pretty familiar with this. And so the cool thing with these liposomes is right now, right, for those of you guys that are going into the medical field or pharmacology, you can actually use this to introduce drugs into individuals, right? So that because they're lipid soluble, they should be able to pass right through the cell membrane without any issues. So now that we have these protobionts forming, got to get some energy, got to have a metabolic pathway. And so if you think back, I mean, we're talking earlier, this is way before oxygen was present. Think about which metabolic pathway that you are familiar with that allows for energy production even though oxygen is not there. And today in us, right, if oxygen is present, not a big deal. It runs with or without oxygen. So think about what type of metabolic pathway that is. So here's where I'm expecting you guys to give me an answer right here. All right. So, but here you can see this little simple pathway, simple metabolic pathway right through here. Did you guys come up with the answer yet? All right, I'm gonna ask you guys on a quiz, I think. So now, in order for these protobionts to replicate, one of the things that we need to take a look at is how can we have, if we have genetic information there, how can we copy that? How can we replicate that? So here's where Cech and Altman come into place. And so these two individuals, they independently was talking about the RNA world. They were actually looking at ribozymes. Okay, so early on, it was an RNA world. RNA is contained in these vesicles, and they ultimately form these protocells. We know that RNA is information storage because we use the information in RNA to actually make proteins today. And RNA molecules, they base pair just like DNA, right? So you guys know that if there's an A in DNA, if there's an A in RNA, there's going to be a U today in RNA, right? C and G, right? So there's the base pair rules right there from RNA to RNA, and then so A to T, C to G, right? For DNA to DNA on there. RNA can also have various catalytic activities as well. RNA is also can be used as a template for to make DNA. So when a virus attacks us, an RNA virus attacks us, it uses the RNA genetic information as a template to make viral DNA. This viral DNA now will then get inserted into your genome, and then now your cells will actually make more proteins for that virus, as well as viral RNA for it. The enzyme that viruses use to go from RNA to DNA, you guys are probably familiar with this, and that's reverse transcriptase on there. So one of the things here is, these guys are most likely thinking, if it's RNA world, but everything today has DNA. You have DNA. You and I have DNA for our genetic information. Your dog, your cat, right? Fish, plants, bugs, bacteria, all DNA. So if it was an RNA world, why DNA today? Okay, so that's what we're going to take a look at. So in order for RNA to replicate itself, Cech and Altman were actually working on ribozymes. Now keep in mind, these guys are doing this independently from each other, right? So a ribozyme, not only does it have genetic information, but it can also catalyze various types of reactions on there. Ribozymes are complementary RNA. They're little short pieces, So when we think about RNA, though, RNA isn't as stable as DNA. So you can replicate RNA pretty quickly, but there's also a higher probability of developing some errors on there as well. Ribozymes contain genetic information, and they're capable of catalyzing a variety of different types of reactions. Now, each type of reaction is going to be very specific for the type of ribozyme that it is. So some of the ribozymes that you guys are probably familiar with are spliceosomes. So if you remember back to your first semester class, spliceosomes are responsible for helping to remove introns, so then the exons can get spliced together. So that's an example of a ribozyme. Okay? And so if we take a look at how proteins are made, and with all the different RNA molecules that are involved with the translation aspect of mRNA, there are some ribozymes that are present there as well. So these are just a couple of examples of ribozymes. Okay. So with an RNA world hypothesis, why do all organisms today use DNA, right? So you guys remember genotype to phenotype, right? That's pretty darn important, right? So DNA, RNA to the proteins, that's a central dogma right there. So one of the big things is we use DNA as information storage today. We all know this. RNA can store that information, but DNA is more stable. And so we can take RNA now, and I mentioned to you guys with reverse transcriptase, convert that back into DNA on there. And one of the things with some metabolic functions is with the catalytic process, proteins tend to be a lot more efficient because, proteins, you're not doing two things, right? So whenever you guys multitask, right, oftentimes it takes a little bit longer to get things taken care of. So with proteins' main role as being catalytic, the nucleic acids now, their main role is to contain the genetic information and the instructions how to do it. They don't have to, now you have one less priority, one less step that you have to kind of deal with. So these are the possible reasons why we have DNA versus RNA as genetic information. The big one is we're actually a heck of a lot more stable, right? It's still prone to mutations, but less prone compared to, say, an RNA molecule on there. Well, this leads us into talking about, some of the early life and some of the early life ideas on there. So a lot of individuals early on, especially prior to Pasteur and him doing a lot of his studies, is the spontaneous generation idea. And that was the main theory that individuals believed on how living organisms came about, right? So here's that biogenesis, life coming from life. And so, but at this point, this is abiogenesis, right? So you have something that was not living, and all of a sudden you got life coming about, right? So they speculated it just spontaneously happened. It's just magic. It'll magically disappear. It'll magically, I guess in this case, appear on there. Abracadabra. So one individual, Francesco Redi, he actually disproved the spontaneous generation idea. And so he did a series of experiments, and what he found was, he had some meat in 2 containers that were open. Flies land on them, they develop maggots, and there are more flies. Now, when he put the meat in the containers and he sealed them, the meat did go bad. However, no flies were inside. Then he did the same thing again, right? So now nothing can pass through. Now he used some gauze or cheesecloth, and so you can still get the airflow going in and out. The meat's still going to go bad, but now the flies can't get onto the meat to lay the eggs. So then notice where the maggots develop. They develop right on top of where the gauze. So this is Francesco Redi's experiment, kind of disproving, saying, hey, life just doesn't spontaneously occur. Well, this was met with a lot of, no, man, I call bullshit. So, little did he know. And then Spallanzani kind of did something very similar to what Pasteur did. So he took some broth, he heated up, allowed it to cool, left the broth open, right? Let the flask open, and he waited, and then found some growth that developed in there. So he took another flask with broth, heated it up to kill anything that was in there, sealed the flask, waited, no growth inside, opened the flask, and then it went funky. Right? So I was like, whoa, wait a minute. So this is kind of like, hey, look what Redi was doing, right? Sealed, nothing. Unsealed, something growing right there. Right? This wasn't widely supported, right? But because some folks, when they did their studies, it's like when you're doing an experiment, right? You and your lab partners, you're doing the same thing. They get one answer, you get something different. Who's right? So one of the things is most likely they probably had some contaminated samples, cross-contamination on there. Right? So one of the things that the early proponents also said, that when you heated it up and you boiled when you boiled your sample right here, you killed off the vital components for spontaneous generation to occur. So that's why we're not going to believe this stuff. So Pasteur's experiment, right? So now we were in 1600s, we were in 1700s, now we're in the 1800s. Okay? Did exactly the same thing this time, but he had a swan neck that was completely open, and so non-sterile liquid, right? Heated it up, and then open air, right? Heated the sucker up, air forced everything out. And so one of the things he did, allowed the liquid to cool over a period of time. There was nothing inside, right? Still was sterile was nice and good in there, but things were settling inside here though, right here where it was open, but they couldn't get it past the swan neck portion. And remember, he actually heated this portion up. And then if he tipped it where the fluid could actually meet up into this swan portion, this S-curve portion of the flask, where the contaminants then could get into the sample, then the microorganisms started growing again. Or not again, but started growing. So if you kind of compare Spallanzani and Pasteur's experiment, they're pretty much the same thing, right? And so both heated them up. One was covered, one wasn't covered. The one that wasn't covered grew microorganisms. The one that was covered, good to go. Kind of the same thing right here, right? Heat this up in this swan neck, nothing can get in there. Still good, but you break that swan neck. Now that it's nice and open, microorganisms can get in there. Now the broth goes kind of funky on there. We're going to change gears a little bit. We're going to move away from microorganisms, and we're going to try to get into some of these larger organisms that you can actually physically see without the aid of a microscope. Okay, and so this geologic record right here, and these various types of periods, eras, and epochs, I'm not going to make you guys memorize all these. You guys don't need to worry about memorizing the dates. You don't need to really worry about when things have appeared. But I am going to go through this kind of chronological sequence. I mean, there are some rough areas, rough ideas that I do want you guys to be responsible for. Okay, but I'm not going to put on the test. All right, during the Devonian, that is known as the Age of Fishes. From what time, what date to what date did the Devonian occur? Yeah, I'm not going to ask you. I'm not going to ask you guys questions about that. I will talk about the Cambrian though, but that's pretty important. I will talk about the Triassic, and so forth. So when we take a look at early organisms, right, and how do we know where things are? How do we know? We look at the fossil record, and with fossil record, there are limitations to it, but the fossil record is pretty darn cool. If you've ever looked for fossils, right? If you go around locally, you can find fossils. You can find megalodon teeth, right? So for those of you guys that watched that movie, The Meg, right? You can find megalodon teeth. I actually had a guy call me years ago, probably about 15 years ago, so yeah, around there, 10 to 15 years ago, and he was asking, he was digging in Mission Viejo. He was digging he was digging in his backyard. He was digging a footing for his retaining wall. He came across this 3.5-inch tooth, and that was about 4 inches, 3.5, 4 inches, and so on two of the dimensions. And so he left a message on my phone. I called him back, and he didn't answer, so I just left him a message. I said, "Yeah, that's a pretty good find. You just found a megalodon tooth." Okay, and so this whole area was all underwater. So I never know what happened with him. But St. Timothy's, when they built the parking structure off of Crown Valley, just west of Moulton Parkway, they actually unearthed some new fossils of whales and dolphins. So that was actually pretty cool to see. And if you actually are hiking up here, you'll find shells all the time as well. And so one of the things here that we'll take a look at is the fossil record helps to preserve, and so we can use that as a dating mechanism of when they were there. But the limitations are the animal has to die at the right time in the right place. They got to get covered. They can't get decomposed. They also can't have, oftentimes soft body parts don't preserve well. It's the hard parts, right? So when you look at Dimetrodon right here, this therapsid, one of the things that you'll see why it's so well preserved is it's all the mineral stuff. It's all the bony material that's actually picking that stuff up. Okay? And so this allows us to kind of date and find when certain things were around. And this was actually a really cool find as well. I mean, they're all kind of cool. So if you go out to the Mojave Desert, so here's where I've taken students out on a field trip. If you notice, this is off of, if you've ever gone out to Barstow, you take I-40 east if you go into the river. And so this is off of Route 66, which is just south of Interstate 40. You can see where our vans are parked, and we basically hiked up through here, and came up right through here, and we went right up here. You can actually see a few people right here looking for stuff. And here's the inset view. You can see this is actually a picture with my kids and another family. They were actually right there looking for trilobite. My oldest daughter, when she was 6, she actually found like 3/4 of a trilobite. So this is very similar to what she found. So this is what they found out there. But the mountains that we were in, we're out near Cadiz and Chambliss, and this is the Cadiz Formation, and it's right here in all this shale, this sedimentary rock, where we're actually able to find all this stuff. So it's pretty cool. So if you ever wanted to do that, sometimes we'll take students out there. We'll go look for fun. But with the radioactive dating, right, the half-life of carbon-14, right, the 5730 years, 5730. All right, 5,730 years. Right? So after that point, you got half, right, and then another 5,730 years. You got a quarter. So you compare the different forms of the isotopes, and you can get a relative age for all these different things. But these are the isotopes and then here's the daughter isotopes. Here's what they spontaneously decay down into, and then so these are the years in which you can actually use this to help give you a rough idea when that particular structure has been found, rock and things like that. The fossil record is also pretty cool because it allows us to document some gradual changes. So if you take a look at horses, you guys are all familiar with horses today. They're really big. They actually stand on their middle finger, okay? And their ring finger and their index finger are actually modified into little splint bones that actually help to support their middle joint. But early on, horses were small in size. They were on 4 toes. Now they're only on 1 toe. So we have all this documented fossil record history, evidence of this. And so horses today are grazers. Grazers are animals that eat grasses off the ground. Right? You're like, what? But browsers are animals that tend to eat leaves that are off of like shrubs and trees. So that's the difference between a grazer and a browser on there. Right? So when you're standing at a magazine stand, well, nobody stands at magazine stands anymore, right? You're browsing through journals. So the fossil record is also pretty cool in that there are transitional forms that have been discovered. And when we take a look at these transitional forms, so you're going to see all these individuals. Some of these cool ones are like Rhodocetus, Pachycetus, and these are that transition from the land animal to the aquatic environment. So these are giving rise to whales and so forth. Right? Pachycetus is actually really cool. So you can see this, and actually, if you go to in the Sahara, there's a place over there called Valley of the Whales, and it must have been like a nursery, almost like a lot of the rocks that you're like kicking around. It's like, people are kicking these rocks, like bowling ball sized rocks. Maybe not kicking them, but they're moving them around. Then they start looking at it closer, like, oh, hey, this is the centrum, it's the vertebra on there as well. But you can see whales actually do have little hip bones, but they don't have hips and legs today. Right? And so when we kind of take a look at some of the comparisons, right? So if we take a look at the fossil record, if we take a look at modern day dogs, and so here's Pachycetus. This is Pachycetus. Right? Pachycetus is this transitional fossil that Philip Gingrich from University of Michigan, he is a paleontologist over there. And so if you look at the name Pachycetus, right, where do you think it came from? It came from Pakistan. Unfortunately, he's not been able to get back over there to do further research. Look at the structure of the bone structure. Compare that to, say, the pig and a deer. Right? They look very similar in structure, and it's quite different from the donk. So we're looking at some of the bones down in the ankle right here. So the earliest, so here's a date that you guys should be familiar with, right? 3.5 billion years ago. The earliest prokaryotes are these stromatolites. So down in Australia, right, they form these little mats. And so one of the things right here that we're taking a look at, so here's 3.5 billion years ago, right? The earliest critters are these prokaryotes, things like related to bacteria and so forth. Right? So oxygen doesn't show up till much later, and it's been proposed that it's been cyanobacteria, right, that were able to produce these. So this is like Anabiana. This is a blue-green algae. You can see what they look like, little mats and so forth. And so as soon as they started using a different type of metabolic process, we ended up with oxygen kind of being given off as a byproduct on there. And then this oxygen revolution is huge because it actually had a tremendous impact on the planet. And a lot of organisms went extinct. A lot of other organisms all of a sudden said, hey, we have a niche that we can fit into. We're going to explode. So we know that oxygen levels kind of changed all throughout Earth's history based upon looking at geologic evidence. So in this pin right here, so here you can see this big pin, you can see all this little red stuff right through here. And one of the things what's happening is the oxygen was oxidizing the iron over there. So now you're starting to get these iron oxides. And so when oxygen first came about, I mentioned earlier, just a minute ago, that it made life difficult for some, while it made life better or easier for others. So by undergoing photosynthesis, you guys are familiar with photosynthesis, right? Now you're using light as an energy source. This also allowed some other organisms to exploit new areas. So now we're going to slowly start to see the transition of plants. Right? We haven't talked about plants. Plants aren't even around yet. We're still talking about blue-green algae. We're still talking about the cyanobacteria. Right? And we're going to see kind of some evolutionary changes that's going to come into play. Right? So over time, we're going to have single-celled eukaryotes, and we're going to have multicellular eukaryotes start to come about. Right? So when we take a look at the eukaryotes, we have an ancestral prokaryote, which has its DNA on the inside, right? And most of you guys remember the nucleoid kind of clustered in a particular region in the cytoplasm. But as the cell gets larger in size, right, that membrane kind of gets a little, it's going to start to fold in on itself. So this is oftentimes referred to as the autogeneous model for eukaryotic evolution, is the membrane starts to fold in as the cell gets larger. And you guys have ever blown a big old bubble that's like the size of your head, right? Notice as the bubble gets bigger and bigger, right, the bubble itself starts to kind of fold and starts to get a little wonky on there for you. Well, kind of the same idea, but remember with phospholipids, right? As they start to pinch in, they can form little vesicles. And so as they start to form these membranous sacs on there, one of the things that we're going to see is the bigger you are, you most likely going to need to consume more energy. So then you start to engulf some smaller microorganisms, and then instead of breaking them down, what do? You keep them around. And what does the little guy do for you? It provides energy for the big guy. The big guy provides protection. So it's almost like the mafia, right? So it's almost like the mafia idea, right? So the big guy provides protection for the little guy in return provides something for the big guy. In this case, it's going to be energy on there. So here you can see on the inside, right? Here is the mitochondria on the inside. Well, there's another line of this where they actually engulfed another smaller organism, and that's the protus. That's the chloroplast. So we have the mitochondria, which is heterotrophic. We have the chloroplast that becomes inside, and ultimately it becomes a plastid, and these guys are autotrophic. And so there's evidence to kind of support this endosymbiotic. So once we get to about right here, where the big cell starts to engulf the little cell, we're talking about endosymbiosis on there, right? And because we're consuming more than one type, right, now we have serial endosymbiosis. And so some of these organelles is what we call them today. They're going to have more than one membrane on there. Notice that the mitochondria has a double membrane. The chloroplast has a double membrane. So if the cell was engulfed again, so as we talk about when we get to protus, and we start talking about some of the protus and so forth, we'll talk about some that actually have more than two membranes on there. Right? But some of them have actually lost this. But some of the other evidence in there is the inner membrane of the cell is similar to the cell membrane. So if we look at the inner mitochondrial membrane right there, that's where the proteins are placed for ATP synthesis, right? Or for oxidative phosphorylation. ATP synthase is there. Oxidative phosphorylation takes place and where we have the electron transport chain, kind of all those things kind of come into play right there. But all those proteins are embedded in the membrane of the cell, the inner membrane. Right? Both mitochondria and chloroplasts, they both have DNA, and their DNA is prokaryotic in origin, right? Small amounts. cell division, they divide upon them on their own. So think about as you're working out and you start to get into better shape, what happens? You see that you place that demand on your muscles, and what do? You end up with more and more mitochondria present, so that way it can supply the energy demands for those working muscles. And then when you stop working out, when at the end of the season, you go into that detraining mode, notice that you get a lot weaker, you can't do things as long, you can't do things as fast, you can't lift as much and things like that, but then you can slowly build that back up again. So they actually will divide on their own. And each one of these little organisms can actually undergo protein synthesis. It's completely different from what the main cell does. And then they actually have ribosomes as well. So there's ribosomes for the big guy, and there's ribosomes inside for the little guy, because they need to make the proteins for themselves. So here you can kind of see this geologic time, and you can see now talk about single-celled. All right, so we talked about prokaryotes, we talked about atmosphere, oxygen in the atmosphere. We talked about, we were talking about single-celled eukaryotes, so that's what we just finished talking about, which might be a good short answer question. Multicellular stuff we're going to take a look at. We're going to talk about some animals, okay? And then colonization of land kind of comes into play after that. So here's the transition from fish, and we have some aquatic amphibians that are slowly going to be starting moving onto land. The ones that don't have to come back to water to reproduce are going to be the reptiles, and then we have mammals that are on there as well. And then here we are, like a second before the stroke of midnight. So as we transition talking about some animals, we're going to talk about the Cambrian Explosion. And during the Cambrian Explosion, this is a particular period that's found during the Paleozoic era. So here's the Paleozoic, and here's the Cambrian. So right here is where we start to see a huge diversification of a bunch of different animals. Now these animals, most of the animals prior to this in the Ediacaran period were more unicellular, but during this Cambrian period, there was a lot of multicellular organisms. Now keep in mind that this is primarily in the marine environment. Okay? Everything's still in the ocean right now. And one of the big things here with this is during the Cambrian, a lot of these organisms also died, but a lot of them had these soft body parts that were actually preserved really well. So when we take a look at the rocks that these organisms were entombed in, right? There wasn't a whole lot of things being shown before then, and all of a sudden, boom, you had all these things, and they died, and they got buried right place, right time, right conditions. All right, so now we got all these fossils to kind of show for it. And so when we take a look at periods of time with no change, the two individuals that kind of proposed the punctuated equilibrium idea was Stephen Jay Gould from Harvard University and Niles Eldredge, they're both from Harvard. Gould has passed away back in 2002, but they collaborated a lot and did a lot of stuff on evolutionary changes. And so the punctuated equilibrium, you can see there's periods of time where there's stasis, nothing's happening, all of a sudden, boom, you got a lot of speciation. Speciation is an event where you end up with a bunch of new species that came about from the pre-existing species on there. And so you have stasis, right? All of a sudden you have big explosion, stasis, explosion, excavation, explosion. And so that's what we're seeing right here during the Cambrian. Well, it's speculated it's during the Cambrian because these are the age of the rocks at which these fossils are actually found. And if you want to read up more about the Cambrian Explosion, you can kind of take a look at it right there as well. So the Cambrian Explosion, we see a large, huge diversification of organisms that weren't seen before. And what helps to contribute to when we see a bunch of new organisms and also we see a bunch of die-offs and so forth, it has to do with continental drift, right? Mass extinctions, and even adaptive radiation, which can lead to speciation events on there. So with the rise and fall of organisms, conditions on Earth were a little bit different. So temperature, atmospheric oxygen, landmass movements, right? We know that the landmass is moving. We're on the west side of the San Andreas Fault, so we're on the Pacific plate. If you live on the east side of the San Andreas Fault, now you're on the North American plate. So most of you guys have probably drove up 15 up Cajon Pass, and as you drive up through there, right, you go right along the San Andreas Fault. If you ever gone out to Joshua Tree and you went out to Keys Point where you're looking down at Coachella, right? If you're looking at Coachella, if you turn to your right, let's say about 2 o'clock, right? So next time you're out there at Keys Point and Joshua Tree, look out towards Coachella, turn so where you can see that Beaumont Pass right there, where the 10 comes through, where you first start to see all the windmills. You can actually see where the Mission Fault and the San Andreas Fault kind of split off, and they actually run together. You can actually see that out there, out in Joshua Tree. So that's pretty cool, right? So these land masses, they're sliding past, right? We know that there's glaciation that happens. I mean, if you've ever been in Yosemite, you've seen glacial. You might have seen glacial polish on there. You might not have noticed it, but if you ever see the shiny part on the granite, that's glacial polish on there. Volcanic eruptions, meteoric impacts, and so forth. So we got a lot of things that actually change the Earth, and that contributed to extinctions and allow for organisms to adapt and radiate. And I'll spend time talking about all of them. So now at this point, we're going to see, early on, right, during the Cambrian, there's a lot of everything's in the marine environment. Now, after oxygen was available, now we're going to start to see plants slowly making their way up onto the terrestrial environment. When we get to the plants, we're going to look at small plants, right? Things like liverworts, hornworts, right? The bryophytes, the mosses. We talk about those, and then we're going to talk. So those are non-vascular plants. We're going to talk about we're going to talk about seedless vascular plants, and then we're going to talk about vascular seeded plants on there. So we're going to spend some time taking a look at plants as well. So one of the things as we take a look at continental drift, right? So this is the plate that I was talking about. We're right here. We're on this Pacific plate, and then here's the North American plate. So we're sliding right along right through here, and then the Mid-Atlantic Ridge. This is actually spreading in all these directions. So as the continents continue to shift, that's going to have an impact on some of the shallow seas, right? Some seas might become shallow, some might become deeper. Areas that were landlocked now might have more effects on there. So with continental drift, what we want to take a look at is the large supercontinent Pangaea. And then as the landmass continues to move, it's separated into the northern portion called Laurasia and the southern portion called Gondwana. And then as we get into more closer to the present day, you can start to see the shapes of the land masses right here. And how did the Himalayas form? Well, the Himalayas formed when the Indian landmass hit the Eurasian landmass, and what do we have right here? Big old gigantic ridge, big old mountain range right through there. Right? So continental drift. So notice that some parts right here weren't exposed to the ocean, so the conditions are quite different. Now as it breaks apart, notice that now you have some more shallow seas present. Right? So temperature is going to be affected. Maybe it's going to be a lot cooler, but the organism adapted to living in a warmer environment on there. So as the land mass moves around, right, the habitat changes, right? And that's where I was talking about the number of shallow water habitats change, right? Climatic conditions change on land. So where do you guys want to live? Do you guys want to live in the 951, or do you want to live along the ocean? Right? So nobody wants to live over there by the San Andreas Fault. It's hot. especially right now, right? It's like 90, 95, 97 degrees out here, and you know, you go down to the ocean, it's like 85, 80, 85 over there. Right? So it's a heck of a lot cooler than it is as you go inland. And then you continue to go more inland, right? It's gonna be like 115. So one of my colleagues, right, she's over there in Cathedral City. I think it's like 115 out there. So as the landmass changes, it also has an impact on the circulation patterns of the oceans, right? And then this is going to allow for what we call allopatric speciation, because now you have a geographic barrier that might have separated the actual original population. So conditions are different in these two areas, and that might allow you to do something a little bit differently. So with allopatric speciation, what we're looking at right here is there is a geographical barrier that separates the population. So in this example, you got this population of trees that can normally exchange pollen and so forth. Now, what happens here is we put a mountain range, or we could put a, you know, you have a big old earthquake, right? And you got a bigger rift in there. So now you have this mountain range right here. Conditions on, say, the north side of the mountain range versus the south side of the mountain range are going to be super different. It's going to be a heck of a lot hotter down here on the south side versus the north side. The north side is going to be a lot cooler. So soil conditions are going to be quite different. So this can cause the organisms that have the better phenotypes to survive that particular environmental condition. So though, remember, genotype determines phenotype, so your genetic makeup. So as long as you don't have any more gene flow between the two groups right here, right, so now you could actually end up with two different populations. And then if they populations do come back together, could they interbreed? Could they hybridize, or do they no longer breed at all? And sometimes you have it where the conditions, the soil conditions change enough where it causes those to become a little bit more different, and there's no longer gene flow between the two populations. This is called sympatric speciation. But what I'm talking about with continental drift and everything is allopatric speciation, where you have a dividing point. That's the big key right there, is there's a geographical barrier on there. So if you think about Madagascar and India, right, they were together before, and now they slowly started to drift apart, and now they're super far apart, so now conditions are quite different. And so if you take a look at frogs, right, this is what we're actually starting to take a look at. Each one develops in a particular environment. They're adapted to that environment. If you bring them together, they may no longer interbreed. There are what we call 5 prezygotic barriers, and there are 3 postzygotic barriers. Postzygotic meaning after the zygote forms. 5 of them, before you can even mate, there are some barriers that come up into play on there. Okay. So with allopatric speciation and geographic barrier, I just want to show you guys some one thing. I can show you more than one thing. So locally, and as we drive north. Right? So you guys are familiar with Vegas. You guys are most likely familiar with Primm, Nevada. Right? So here's Needles, and here's LA. So what we're going to come through, here's Barstow. And so as we drive through right here is the San Andreas Fault, runs right straight through here. We're going to go up I-15. We're going to go out here to Barstow. Right? We're going to go out here to Baker. Right? And then we're going to go out here go over the last hill, drop down into State Line. So here's the 15 right through here. Right here, I-15 up to Vegas. And before, this was all interlinked. There's a large lake and tributary system that comes into play. And so what we have is what they call desert pupfish. And these desert pupfish are isolated now in these various pools, because a lot of these lakes now are no longer interconnected. China Lake is, if you're ever going up the mountain, China Lake, that's a dry lake bed. Owens Lake, right? You don't really see Owens Lake. You see Mono Lake. Mono Lake is way up here, right? Then you'll see Crawley and so forth. But like Searles, that's a dry lake bed. There is no Panamint Lake. Lake Mannix, out here, that's a dry lake bed. Manly, that's a dry lake bed. So there are some small remnant pieces of this, and you find these deserts, and each one is adapted to its specific environment. There's actually a story where there's the Owens River pupfish. Phil Pfister was a USGS fisheries biologist, and it was bad back in the 80s and the early 90s. So he had 2 5-gallon buckets of the Owens River pupfish, and he was walking back to his truck thinking to himself, I just collected all of the pupfish, Owens River pupfish. If I trip and fall, the extinction of this species is all because of me. Right? So he was like, oh my gosh, what a tremendous weight that was on his shoulders. Obviously, they got him to an area where the Owens River, it did not dry up as easily. And so this one right here is actually out in Death Valley right here. This is the Death Valley pupfish. There's actually another one the one that's really is Devil's Hole, which is close to Beatty. It's actually right about here. Okay, Lake Tacoma, Shoshone. Yeah, it's actually right about over here, right near the state line, right there. So we have continental drift, and then we also have extinction, right? Mass extinction. So as soon as you have a bunch of die-offs, what can happen? There's a lot of job openings, right? There's a lot of niches to fill. You can have organisms kind of come in and fill those particular areas. So climate change has contributed big time to mass extinction. And so we're actually looking at that right now, is there are organisms that are slowly starting to die out. We know that there are some tree species down in the Andes that, because of the climate's getting warmer, these trees are actually normally found down in the lower ranges, but they're actually, because it's getting warmer at higher altitudes, we're actually starting to see these trees now up at the higher altitudes. And then this is having a huge impact on the trees that are normally up at the higher altitudes, right? They can't compete as well with these trees that are adapted to that warmer environment. So now we're starting to see them slowly start to die off, right? So there are major geologic events, right? Continental drift, earthquakes, mountain formations that have a huge impact. And you have a ginormous asteroid that impacts you that can kill off a whole bunch of things, where you have this big old cloud and it becomes where the temperatures drop because of all the particles that are up in the air, covering the sunlight, available sunlight that can hit the earth. Right? But there have been 5 major mass extinctions on this planet where we see a bunch of organisms die off. Right? And most of these are, again, like these first 3 right here, they're mostly in the seas. Okay? And this one right here, this is the one that got the dinosaurs right here. So a major mass extinction over here. But the one between the Permian and the Triassic right here, right? So right here, what we're looking at right here, volcanic eruptions, global warming, ocean acidification, and decrease in oxygen availability coming into play. Okay, and so notice how these changes can have a huge impact on life on Earth. So here's the Cretaceous mass extinction. That was the one. Right? This is what's speculated. And so now that you have these die-offs, right? Notice that we can have increased numbers of new animals, right? New species kind of come into play, right? So we start to see more and more. So here's the one where the dinosaurs. Now, in addition to that, some areas, right, you might not have had any predators. If you have soft bodies now, there's a bunch of organisms that have soft bodies. Now you got some predators that have some different types of armament on there. That creates a big disadvantage to say the prey item. So there's a lot of competition and there's a lot of selection that kind of comes into play on there. Okay, especially after that. So what I want to talk about with you guys now is what we call adaptive radiation. Okay? And because the dinosaurs are gone, the mammals now start to, hey, some of us don't need to hang out at night anymore, right? We can become active during the daytime because we know the dinosaurs are active during the daytime, right? And so if you think about mammals, when are most of the mammals active? At night, right? Why are they active at night? It's because you don't have all these big, large, terrible lizards running around in the evening. Why? Because it's colder during the evening. So notice that most mammals have a lot of fur, except for us. Right? So following mass extinctions, right, into organisms with some unique characteristics. Novel refers to unique or new characteristics, and this could be the result of changes in selection. And then so adaptive radiation, we have a group of organisms going to a new environment and adapting to that given environment and being able to take hold in that new environment. So when they spread out, that's the radiation part, and then they have to adapt to that particular environment on there. So notice that we have three different types of mammals. We have egg-laying mammals, we have marsupial mammals that have a really short gestation period, and then we have the eutherian mammals, the true mammals. These are the placental mammals on there. So with adaptive radiation, what we're looking at, we're talking about colonization. So here is the mainland over here, right? Species A now gets over here to a nearby island. And so now here's the radiation aspect of it. And all the conditions over here on this nearby island are going to be different from the mainland over here. And so as a result, some of the species A is probably going to die off, but some of them are going to be able to adapt because they have the better phenotypes that fit for that particular environment. And as a result now, these guys, if they come back over here to the mainland, they may not be able to interbreed with species A. But now species B now actually, because of competition, right, it's getting crowded, they'll move to neighboring island. And then conditions are different from number 3 versus number 2. So then they can actually move back and forth. So now notice that South C moves back over here to this first island, but they don't interact with each other. And as a result of that, now, it's maybe because different song, right? Different mating season, different mating time of day, right? So if those three things, right? If you want to have a baby, you got to be at the right place. Your timing has to be right. And then parts have to fit on there as well. So you got all these things that kind of come into play that could prevent species from interbreeding with each other. Right, so that's how we maintain that species barrier on there as well. So, and you can kind of take a look. So this is the explanation for the Darwinian finches on there, right? And because of mass extinction, there are new job openings. I like to think of niches as a job opening, right? You guys are getting educated because you're trying to find your niche. Right? You're trying to get all this stuff so that you can fit into a particular niche. Create a niche for yourself. Right? So we can go to Hawaii. Islands are the greatest places to look for adaptive radiation. So here's these tarweed, but you go to the different islands, right? They're adapted to the different conditions that are there, right? Some are growing taller, some are growing shorter, some are more tree-like, some are more shrub-like, and so soil conditions are different. The amount of rainfall is quite different. The exposure, north side of the slope, south side of the slope, and so forth. Okay, so a lot of, the wind, all these things kind of come into play. So as we take a look at the rise and fall of animals, what I want to talk about now with you guys is looking at the development and the timing. So when we take a look at the changes in body form, one of the first things I want to take a look at is what we call heterochrony. Hetero refers to different, chrony refers to timing. So the developmental rate or the timing of the development can vary from species to species. So you can have more of a particular stage, it could take longer to get there, more stages but at the same rate, or you could actually even start earlier. So if we take a look at the newborn, right, we grow pretty quickly early on, But then as we start to get older and older, our growth rate starts to slow down. Right? Because think about when you're a kid, right? You have a ginormous head and your body is teeny tiny, and you run like, you know, see, obviously you can't see me kind of pretending to run, right? You run with your head forward, and then that's why you trip and you end up with a taller tattoo with a big old knot on your forehead on there. But so if you look at the chimpanzee and the human, right? The skulls, the fetal skulls are growing about the same, right? But when they're born, right, growing a little faster, and then but our skull continues to grow even after we're born. We can take a look at zebras. Look at the striping of the zebras. Right? This has to do with the timing of which. So we have Burchell zebras, we have Grevy zebras, and we have the mountain zebra. So these are 3 different species, and then you got a couple of subspecies of Burchell's zebras on there. Right? But look at the striping pattern on there. So we're talking about, you know, some of the stages running faster, some of the stages running at a shorter time frame, and some of them you have a couple of them that are running at the same time on there. So heterochrony is what I want you guys to understand about heterochrony is it's looking at the developmental rate or the timing of the rate. You know, is it running faster? Is it running slower? Some organisms now are what we call paedomorphic, and this looks at the timing of which. Okay? And so with organisms that are paedomorphic, they become sexually mature in the larval stage. So they're sexually mature, but they still show larval characteristics. So this is the Mexican tiger salamander. It's actually called the exotle. It's actually really unique. It's traded widely in the aquarium industry. This is the larval salamander condition still has gills. They actually have lungs too, right? You can see the webbing on the tail, and so one of the things here that helps the transition is not temperature, but also hormone changes as well. So these guys are actually pretty endangered now because they're being widely collected on there. But this salamander is actually sexually mature, even though it has larval characteristics on there. Right? So the reproductive rate occurs at a much faster rate than in which the body is developing on there. So, and I put these 2 on there because this is the genus is Ambystoma. The species, the specific epithet is mexicanum. This is Ambystoma tigrinum. This is Ambystoma mexicanum. This is Ambystoma tigrinum. Right. So this is the tiger salamander. This is the adult form, and it will undergo metamorphosis. And so now it loses its lungs. It loses its gills, but now it relies solely upon its lungs. Not solely upon its lungs, because amphibians do a good bit of their gas exchange across the skin cutaneously. Okay. And so neoteny, you might come across that term. That's so this is an adult, and this is referred to as neoteny because this is an adult salamander, but it retains juvenile characteristics, and it's also sexually mature on there. So the timing of the reproductive rate compared to the body development. The other big thing that affected a lot of organisms and their changes in the body form has to do with this spatial pattern. We have what are called homeotic genes. All vertebrates have these homeotic genes. All animals have these homeotic genes. And this has to do with the placement and the organization of your body parts. So our body is divided into segments. Right? So we have 3 main segments. We have the head, we have the thorax, which is your chest area, and we have the abdomen on there. Right? And so within these homeotic genes, we have specific genes in there that are called Hox genes. Okay? So these homeotic genes deal with the placement and the organization of the body parts. The Hox genes now deals with the positioning. And so if we kind of take a look at this lancelet right here, one of the things when we take a look at this, you can see all the different Hox genes that are present. So when we take a look at the duplication and we undergo the copying, you can actually double the number of genes that are present. And is that an error? Yeah, if the error is favorable, sure, that's great. And then as we continue, one of the things that we see as we continue to go up to the vertebrates, we see more and more of these various types of Hox genes. And in some of these, we actually see duplications of these particular Hox genes. So now what comes into play is you have these genes that are present, homeotic genes and these Hox genes. The homeotic genes and the Hox genes now. So remember, the Hox genes are within the homeotic genes. And so these Hox genes, there are certain regions that get turned on and turned off. And one of the things that they can do is, if they are turned on, they might express a certain amount of stuff. So how long are they turned on for? So here, what we're looking at in the chick embryo, we're looking at the thorax. So this is like the thoracic region right through here. Right. Here's the thoracic region right through here for the snake. So notice that we have this expression. Right? This expression is on for a longer period of time, and you end up with more of these thoracic vertebra on there. So the thoracic vertebra, you guys all know, contains ribs, right? So here's the thoracic vertebra here, here's the ribs right here, here's the thoracic vertebra, look at all the ribs right through here. Okay, so we're looking at the timing of which for these two reptiles. So now, again, with this, each Hox gene can, our whole body, these homeotic genes are divided into segments, and each Hox gene controls a particular segment. So how long you turn one on will determine how much of a structure you can actually produce. So here we have brine shrimp right here, and then notice that it has a lot of little swimmerets. Right? Here's a Drosophila, there's a little fruit fly. It only has, so this one isn't turned on. It's turned on really quickly and develops very quickly, but then it's turned on. But this one has a longer duration, right? So where it actually starts, right? So it's in all these different body segments. So in each one of these body segments, here's where that gene is actually turned on. Whereas over here, it's only turned on in three of these body regions right here. Whereas more in the Now, I'm not going to spend a whole lot of time talking about evolutionary novelties in the eyeball, but the eye itself, photoreceptor cells in the eye, pretty darn complex. And there's some convergent evolution between the eye of a squid and our eye on there with the lens. And what you guys will have an opportunity to dissect an eyeball this semester as well. So here's this geologic table again. Again, you guys are not responsible for memorizing anything on here. I just have it up here so you can kind of have a rough idea of when certain things come about. You know, and it's actually pretty cool stuff. So a lot of you guys might be traveling right now, and if you're going to these different areas, there's a lot of cool things that you can actually see out there, and it ties in with this geologic table. So if you have any questions, feel free to shoot me an email, pop into office hours, and I look forward to helping you guys out, talking to you guys soon. All right, have a good one. Bye now.